Chapter 23 is one of these fun chapters where we get to learn about all of these specific diseases that you may see out in the field. Chapter 23 specifically talks about parasitic diseases. So when we talk about parasitology, we are very specifically talking about either a protozoal disease or a helminth disease. These are both eukaryotic parasites. They're like the larger parasites. So first, I'm going to discuss the protozoal diseases, and then we'll get into the helminths, the worm. Now, some quick facts about these parasitic diseases. They cause about 20% of all infection worldwide. But the thing is they're much less prevalent in industrialized countries. So although there are definitely some that are endemic, even to the United States, they're more likely to be found in tropical environments, mostly because a lot of these diseases spread by very specific insects or arthropods that are only found in very tropical locations. Another reason why you're less likely to find them in industrialized countries is because we have very strict water treatment protocols. Several of these, not all of them, but several, especially the protozoa, can be spread via drinking contaminated water or water that's contaminated with fecal matter. So in the United States, we usually have very strict water treatment protocols. In addition, we have very strict food processing protocols. So we are more likely to screen our meats and our animals for these infectious diseases. And also we know to cook our meat thoroughly. Another thing is in our seafood preparation, in the United States especially, we're very likely to freeze our seafood before serving it, and that's one way to help prevent diseases due to the eggs of helminths. Now, where these diseases are really significant is in AIDS patients. Because they are immunocompromised, these diseases can progress very rapidly and lead to severe secondary infections. So like I said, first we're going to talk about the protozoan pathogens. And I just want to remind you, protozoa life cycles are often very complex. Not all of them, but many protozoa form a cyst stage, which is basically like a dormant stage. And this is usually the way that the protozoa enters into the body. And then once it gets in the body, it can activate into the trophozoite stage. So you can think of the trophozoite stage as being the actively feeding and replicating stage. Now, there's a couple different ways we can classify protozoan pathogens. A lot of medical textbooks will classify them by the type of organ system they infect, like whether they infect gastrointestinal, blood, tissues, etc. Our textbook actually classifies them on their type of motility. Remember, amoebas have those pseudopods, ciliated protozoa, which have those small hair-like extensions, the flagellated protozoa, which have a flagella, and then lastly, we're going to talk about the apicomplexan protozoa, or the ones that don't have a form of motility. In this first video, I'm going to talk about the amoebas and the ciliated protozoa. The first protozoa disease belongs to the amoeba class. It's called Amoeba histolytica. So when you see this, you can see it's italicized. Amoeba is the genus name of this organism. Histolytica is the species. Amoeba histolytica causes a disease known as amoebiasis. So amoebiasis is the disease this organism causes. Now, Amoeba histolytica could be a sporadic disease, as with a lot of these gastrointestinal protozoa. They can be sporadic, meaning we're more likely to find them in institutional settings, such as maybe daycares, where a lot of children are in close contact, or prisons, or military barracks, where people are consuming the same types of foods and they're in close contact. These gastrointestinal protozoa may also be endemic, meaning we're more likely to find them in certain regions in the world, and usually, like I mentioned, more tropical regions. Amoeba histolytica actually causes what we call dysentery. Whenever you see dysentery, a lot of these diseases cause dysentery. That indicates one of the symptoms is a bloody diarrhea. Now, how this disease spreads is normally somebody will ingest the cyst of the Amoeba, usually in food or in water, Once the cyst reaches the intestines, it can form the trophozoite stage. And in the trophozoite, if we look at it under the microscope, what we'll see is actually the human red blood cells will be inside of the trophozoite. So obviously the number one way this disease spreads is through ingestion of contaminated food or water that contains the cyst of the entamoeba. However, another way that this disease can spread is actually through sexual contact, particularly men who have sex with men. Once the trophozoite enters the intestines, it actually from there releases a cytotoxin. And this cytotoxin slowly destroys and damages the colon epithelium. However, it has been estimated that almost 90% of individuals infected with this disease are asymptomatic. Occasionally, the trophozoite can actually enter the bloodstream, and where it will target is the liver. It can immediately enter the liver. A lot of patients will complain of having right upper quadrant pain due to the entamoeba targeting the liver. This picture is just to show you what that erosion of the intestines look like. As I mentioned, the cytotoxin, which targets and slowly destroys the intestine. Most patients, as I mentioned, will be asymptomatic, but some will experience that bloody diarrhea, abdominal pain, fever, weight loss, and like I mentioned, if the protozoa reaches the liver, they might feel pain in the upper right quadrant. To diagnose these intestinal protozoa, we have to take a stool sample, and we have to actually look for the cysts and the trophozoites in the stool. The problem is these protozoa don't always show up in the stool sample, so we always take 3 separate samples. What you would likely see under the microscope is the trophozoite very clearly is going to have 4 nuclei. That's like a characteristic feature of the Entamoeba histolytica. These 4 nuclei, 1, 2, 3, 4 that are inside of the cell. You may also see, again, red blood cells within the trophozoite cell. One important place you may see this in the field is from a traveler. If someone who's recently traveled outside the country, they come back and they're experiencing dysentery and upper right quadrant pain, then we might take a stool sample and look for the entamoeba. Treatment is often metronidazole. Metronidazole is a very commonly prescribed drug for these anaerobic protozoa. Also, iodoquinol is an effective drug for Entamoeba histolytica. There are 2 other amoebic protozoa. These 2 actually don't cause gastrointestinal issues. They actually target the brain, and they can actually lead to meningitis or protozoal meningitis. So these 2 are very severe pathogens that we would really be concerned about. The first one is Naegleria fowleri. Now this one is one we usually see in the summer month because this protozoa lives in standing water. So for example, lakes or maybe a swimming pool that's not properly chlorinated. Although some of the protozoa may not be affected by chlorine at all, so it might even be able to survive it. Now, I don't want you guys to all freak out like, oh, I can never swim in a lake again, because a lot of people come in contact with Naegleria fowleri in the lake while they're swimming, but they don't get it. But it's very rare that someone actually progresses to an infection. But when it does progress into an infection, about 95% of people will die within 1 week, as the protozoa will usually enter through nasal passages while swimming, and then it can enter the brain and the spinal fluid. Typical signs and symptoms of a Naegleria fowleri infection are the same as a bacterial meningitis. So the patient will feel a fever, headache, stiff neck, nausea, vomiting. And a lot of doctors don't know whether it's this protozoal meningitis or bacterial meningitis. The only way to tell is by taking a sample of the cerebral spinal fluid and actually look for whether it's a protozoa there or bacteria. There has been, I think, 2 people that have survived this disease, and they both took this medication called amphotericin B. The second amoebic infection of the brain is acanthamoeba. This one is less dramatic. It's more likely to only affect someone who's immunocompromised or has AIDS. And it actually also has been associated with contact lenses. So you never want to put contact lenses in just plain water because there could be this amoeba in the water. And I think there has been a time where the contact lens solution actually became contaminated on the market. They had to do a major recall. So if this protozoa enters into the eye, it can actually lead to blindness as well. So those are the amoebic protozoa. Now there's one ciliated protozoa. It's another intestinal protozoa. It's actually called B. coli. You've probably heard of E. coli bacteria, but this is a B. coli. It's a ciliated protozoa. Now this one spreads from the oral to fecal route, but it very specifically spreads through pig feces because this protozoa is normally found in the intestines of pigs or cattle. So how this may happen is, for example, if there's pigs living and residing on a farm that also grows lettuce or fruit, and the pig feces contaminates the produce, and then somebody doesn't wash their produce properly and they ingest this protozoa. Or if there's a water stream nearby and somebody drinks from it and there's pig feces in there. Now, similar to entamoeba, the individual will usually ingest the cysts of the B. coli. The cysts will mature into trophozoite and they'll travel to the intestines. Now, what's interesting about this protozoa is it feeds on the normal flora. So these guys will happily start growing in the intestines. They'll feed on the normal flora bacteria in the intestines. And as they feed, they're actually digging and eroding the intestinal cell wall, and that's what causes symptoms. Many people are asymptomatic, but some people will experience diarrhea due to the infection. Interestingly, this is the largest protozoa there is. And again, the way we would diagnose this is we would collect 3 stool samples, look under the microscope for these large ciliated protozoa. Treatment is typically a tetracycline. Tetracycline is an antibiotic, but it actually also will destroy this ciliated protozoa. We may use iodoquinol again or metronidazole.